It's a pleasure to introduce our last speaker in this panel. Robert Kass, Rocky Kass, is Vice Dean of Research at Columbia University and the Chair of Pharmacology. He has a long history of working both in physics and as a physiologist. He's published many papers on properties of different ion channels and their pharmacology, in particular sodium channels and sodium channel antagonists.
Thanks, Mark. Well, it's a pleasure to be here. I want to thank you for inviting me. And now I'm going to, like Monty Python said, now for something completely different. We're going to work on function. And I'm going to focus on cardiac electrophysiology. So I'm going to give you a little teeny bit of review. And the goal of the talk is to show you how we've used iPS cells induced into cardiac myocytes to correlate and improve the treatment of a particular patient. So this is patient-specific use of iPS cells. But in order to understand what we did, I've got to give you a little bit of background about the electrophysiology. So I'm sure all of you know that during each cardiac cycle, there's a regular and repeated rhythm that's coordinated by electrical signals, where there's a pacemaker region of the heart where a group of cells called the sinoatrial node fires and drive impulses through both atria, throughout both of the ventricles, and excite the ventricles quickly to contract. Each of the cell types that have these different physiologic functions have different signature waveforms, if you will. And we're going to be focusing mostly on the electrical activity of ventricular cells, which typically in the heart lasts a very long time, has a very rapid upstroke, and has a period between depolarization and repolarization, which I usually call the plateau phase of the electrical activity, because there's very little change in activity versus time. You all know, I'm sure also, that clinically the measure of this electrical activity during each cardiac cycle is the electrocardiogram, which is very important and very useful for the disorder that I'll be talking about, because it gives a non-invasive measure of a patient's electrical activity. And the electrocardiogram, the ECG, has different waves that have been named the P wave, which corresponds to the excitation of the atrial muscle, the QRS complex, which is the very rapid depolarization of ventricular cells at the cellular level. That's reflected in this very rapid upstroke of the cellular action potential. And then there's this sort of quiet period between the QRS complex and what's called the T wave, which really reflects this plateau phase of the cellular cardiac action potential. And it's just the time between the excitation and relaxation of the ventricle that I'm going to be focusing on. And you'll hear me over and over talk about the QT interval. And that's because the disorder that I'm interested in today and have been interested in the lab for many years is referred to as the long QT syndrome. As the name tells you, it's a syndrome in which the QT interval of patients is longer than a certain normal period. The ECG, don't forget, is electrical signal versus time. Prolonged QT means there's a delay in repolarization in these patients' hearts. By itself, delay in repolarization, prolonged QT, pardon me, is not particularly problematic. In fact, in the 80s, drugs were being developed to prolong QT intervals to prevent certain kinds of electrical disturbances called reentrant arrhythmias. But what they found, what drug companies found in the 80s and early 90s, was that prolonged QT often led to very dysfunctional and often fatal electrical activity, arrhythmias in the heart. And so it's the link between prolonged QT and dysfunction and sudden electrical cardiac death that we're very interested in understanding and preventing. And as I said initially, this sort of prolonged QT could be, and still is, induced by drugs. That's called the drug-induced long QT syndrome. But the syndrome that has continued to give us the most information about the mechanisms that underlie this dysfunction really is the congenital long QT syndrome. And this is the fact that this disease is now known to be caused by mutations in several different genes, about 12 to date. And all of these genes either code directly for ion channel alpha or beta regulatory subunits, or adapter proteins that interact with and modulate ion channel activity, or direct ion channels at different parts of the cells. Despite the fact that there are 12 now genes that are known, they're really four primary forms of the long QT syndrome. And if you remember from basic cardiac or basic cellular electrophysiology, anything that prolongs an electrical response, prolongs an action potential, would come about at the cellular level by something that would reduce repolarizing currents or increase excitatory currents. The repolarizing currents in the heart are known to be potassium channels. The drug-induced long QT syndrome turns out to be due to drug block of a particular potassium channel, which I'll come back to talk about. And it turned out that the first two major forms of the long QT syndrome, long QT1 and long QT2, were discovered to be due to loss of function mutations in genes coding for primary potassium channel alpha subunits in the heart. KVLQT1, now referred to as KCNQ1, makes up long QT variant 1. KCNLQT2, rather, is due to the hERG gene, which is actually the same gene product which is the target for drug-induced long QT syndrome, a fact that we're going to come back to importantly later in the talk. Surprisingly, to me as an electrophysiologist, and probably the most interesting, were the mutations that were discovered in the gene coding for the cardiac sodium channel, SCN5A gene, that codes for the primary sodium channel in the heart, which is thought physiologically to be most importantly responsible for rapid impulse conduction through the heart. So how can a sodium channel mutation prolong the action potential at the cellular level and the QT interval at the systems level? Remember, sodium channels turn on very rapidly and generate the rapid upstroke and rapid conduction during the QRS interval. And normally sodium channels, once they open, they enter a non-conducting inactivated state and are normally not important to controlling electrical activity during this period. But the first long QT3 mutation that was discovered turned out to be discovered in an very interesting area of the channel, an area of the channel which we, by people who are studying biophysics, knew was responsible for generating this non-conducting inactivated state. It was a peptide loop joining two different domains of this alpha subunit of the sodium channel. The peptide loop is called the inactivation gate. And the inactivation gate normally physiologically, once channels open, sodium ions rush in, give you the QRS complex, spread impulses quickly. This 3,4 linker occludes the pore, blocks the pore, and prevents sodium ions from coming in and gives you the non-conducting sodium channel activity during the prolonged QT interval of the electrocardiogram. The mutation is a deletion of 3 amino acids in the inactivation gate. And what's great, what I like about these sorts of experiments, is that you can see clearly functionally there's a difference between the activity of a channel that includes a disease-causing mutation, in this case the delta KPQ mutation, versus a wild-type channel. I'll be showing you some records, and the records I'll show you will be at high gain because these this sort of residual sodium channel activity, which you see compared to the baseline zero current here, this inward current during this prolonged time during depolarization is what underlies the long QT3 version of the disorder. That inward sodium movement is what prolongs the action potential, prolongs QT, and leads to torsade de pointes and sudden death. So the goal, maybe as long ago as 8 years ago, was to determine whether or not there were drugs that were usable within the clinical toolbox that might preferentially inhibit this dysfunctional late sodium channel activity without doing much to this peak sodium channel activity, which is, I would call this the voltage clamp equivalent at the ECG. This is the current underlying the QRS complex. This is the QT interval prolonger. You don't want to do much to the QRS complex because you don't want to slow impulses when the ventricles are excited, but you want to inhibit preferentially this late sodium channel current. And it turns out that there's several drugs that are pretty effective at doing that. Mixilotine, lidocaine, flecainide, and a newer drug, ranolazine. And in all of the work that I've done, we've closely collaborated with clinical colleagues who are treating patients who are genotyped for this disorder. And Arthur Moss in Rochester is a longstanding collaborator of mine. And Arthur quickly tried some of the drugs that we were finding at the cellular level were effective at correcting this dysfunctional sodium channel activity. Here we're looking at the effects of flecainide on a long QT3 patient. And all I'd like to show you is that the QT interval, which in the absence of drug is prolonged and is within the range of long QT syndrome, in the presence of this sodium channel blocker, which at the cellular level we know inhibits this late sodium channel current, corrects the QT interval with minimal effects on the QRS complex, exactly as predicted from the cellular in vitro experiments. So we were really happy. We were thrilled. We had a mutation-specific approach for treating long QT3 patients and had a selection of drugs that were preferentially inhibiting this dysfunctional channel activity. The problem was that this approach didn't always work. And a case came to us at Columbia where there was an example of a newborn who was born with incredibly prolonged QT intervals. This is a controlled baseline electrocardiogram from the child at birth. His QT interval was on the order of 800 milliseconds, which is very, very, very abnormally long. And the clinician working on the child collaborated with us and asked what kinds of drugs might we screen. And we suggested trying drugs that might be useful for inhibiting this late sodium channel activity or drugs that might be preferential for potassium channel mutations. And he found that lidocaine, one of the drugs that we knew ought to correct this dysfunctional activity, was very good at controlling this dysfunctional electrical activity and converting it into normal rhythm. The problem was that this normal rhythm couldn't be maintained, and the child deteriorated into torsade and to serious arrhythmias frequently, regardless of the sodium channel blocker that was used. So an implantable ICD was put in to protect the child, and then we, in collaboration with with Wendy Chung, who's a molecular geneticist in pediatrics, genotyped the child and found, in fact, he was carrying a mutation right in the same part of the channel as the original delta KPT mutation, the inactivation gate of the channel. A single amino acid change. This was a spontaneous mutation. Neither parent carried it. When we looked functionally at the impact of this mutation on the sodium channel, you can clearly see this dysfunctional activity, which is apparent in the channels where the mutation is included compared to wild-type channels. This is DNA that's expressed in a heterologous system, HEK293 cells, and that's this very, very large late sodium channel current is clearly much more marked in the cells where the channels contain the mutation versus control. When we screened for drugs that we thought might be useful, again, this is in the heterologous system, we looked at flecainide, we looked at mixilotine, we looked at ranolazine. They all preferentially inhibited this late sodium channel activity with little or no effect on the peak sodium current, which suggested that they all should have worked reasonably well in this patient, but they didn't. And so we wondered what we might still be missing. And it's at that point that we went back and looked more carefully. These are records showing that the child, while under reasonable control, would deteriorate frequently, even in the presence of one of the sodium channel blockers, which was mixilotine. Turned out that not only did the child have a spontaneous mutation in the sodium channel, but the child was born being heterozygous for two forms of this important potassium channel, the KCNH2 gene, which codes for the channel which is responsible for drug-induced long QT syndrome. And we speculated that perhaps the fact that the child was heterozygous for two variants of this potassium channel, as well as carrying this very strong disease-causing mutation in the sodium channel, might compound the pharmacology and the therapeutics of treating the child. And so we decided to collaborate with others to see whether we could make iPS cells from the family and study iPS cells that were induced to differentiate into cardiac cells to look at the in vitro pharmacology, and then compare that with what the treatment for the child was given by his cardiologist. We collaborated with two different labs. We were working with Darrell Cotton at Boston University, who used the Yamanaka cocktail to make iPS cells. We then collaborated with Gordon Keller, who's now at the University of Toronto, and Gordon worked with the iPS cells to differentiate into cardiac cell lineages. And we wondered whether or not we would first be able to detect dysfunctional channel activity in these cells, and then whether or not the cells would be useful to screen for drugs so that we could then go back and recommend differences in approaches to the clinician managing the case. So these are the first recordings that we made from these cells. And what we did is we imposed voltage clamp recordings of action potential waveforms. These are the action potentials you would see from a normal adult ventricular muscle. We blocked all channels but sodium channels in these recordings. And you can see the wild type or the normal channels turn on very quickly, underlying this rapid upstroke, and they shut down as we might have expected. And we were very, very excited because when we looked at the cells derived from the proband, we could see very dysfunctional sodium channel activity. We carefully characterized the sodium channels that we saw in the normal iPS cells, and we were lucky enough to have some data from human HES, human embryonic stem cells, that we could compare them to. And we found that the sodium channel activity that we recorded from the normal cells, regardless of what clonal line we looked at, were identical to the sodium channel activities we saw in normal HES2 cells. This is the sodium channel availability curve. I'm not going to talk very much about that, other than to say that the channel activity that we saw in the normal cells agreed exactly with the channel activity that we saw in the HES cells. And fantastically, we thought, when we looked at using the same protocols to compare normal channel activity with channel activity from the proband, we could clearly, very readily see this very pronounced dysfunctional sodium channel activity in the iPS cells. So step one is that the myocytes derived from the iPS cells can be used to study dysfunctional channel activity that underlies human disease. We can characterize the biophysical properties of these channels in a way that we never had been able to do before. And then the next step was to ask whether or not this was a consistent, whether these properties were consistent, regardless of the clonal line. We have 2 or 3 additional lines that we've since characterized since this slide. We always see this very pronounced late sodium channel activity that we don't see in the normal cells. Importantly, this steady state sodium channel availability curve is also affected by the mutation in a way that is also pro-arrhythmogenic. Because these are cardiac cells that are derived from the iPS cells, the advantage they have over expressing channel activity in heterologous systems is that all or most of the channels that are involved in generating the cellular activity are available for examination. And what we wanted to focus on, remember, was the potassium channel, which had, where the child was heterozygous and both parents were homozygous for 2 different variants of the channel. And that's this KCNH2 or IKR channel. These are just different recordings from the different cell lines. They all look roughly the same. They all have the same biophysical properties, and that they're detectable, they're reproducible, but the biophysical properties of the channel were not different in the proband versus the parents. And so that the hypothesis that we had that perhaps the fact that the child was carrying was a heterozygote for 2 different forms of this channel didn't appear to be related to the problems in controlling his arrhythmias. We then looked at drugs. We chose to look at mixilotine because mixilotine was the drug of choice by his physician. We looked at mixilotine on normal channels and just characterized the concentration dependence of block of the very peak current, which we don't want to be blocking very much as we treat the child. And since there's no late current, there's no late current to block. When you look at the dysfunctional channels that we could readily see in the iPS cells derived from the proband, mixilotine very potently inhibits this late channel activity, and it's much more potent on late versus peak, which is exactly what we wanted to see. The other caveat here is that in order to control these arrhythmias reasonably, the clinical dosage of mixilotine was high. That's a point that we need to be coming back to. The concentrations that we were using here were comparable to the concentrations used to treat the child. But what we found, because we had these other channels that we could look at in the iPS cells, was the same mixilotine concentration that was antiarrhythmic, that was blocking this late dysfunctional sodium channel activity, turned out to be inhibiting this potassium channel, which by itself would cause drug-induced long QT syndrome. So we had a mixed effect of a drug. We had an on-target effect, which we wanted, inhibition of the dysfunctional sodium channel, off-target effect, inhibition of this potassium channel, giving competing actions at the cellular and competing actions at the patient level. The antiarrhythmic effect of converting the dysfunctional sodium channels into normal-looking sodium channels, but the unwanted effects of blocking the potassium channel, which would cause a prolongation of the action potential. And so just as we saw, even though mixilotine was useful at controlling the rhythms of the child, often we would see the child go into torsade and be corrected with the implanted device. So we asked, are there other things that we could have looked at and could have done to improve the therapy of the patient? One approach would be multiple drugs. A number of the clinical colleagues of ours suggested that. They raised the possibility of using mixilotine and flecainide. I'll show you how we tested for that. But we also looked at the idea that maybe there was something simpler we could do. Because there was an implanted device, the heart rate of this proband could be changed. And it turns out that if we look at the risk factors involved for this form of the long QT syndrome, long QT3, we find that that we're looking here at the percentage of patients who were genotyped to 3 different variants of the long QT syndrome, the percentage of incidents where cardiac events occurred either when the patients were exercising, when they were startled, or when they were resting. And so when they were resting, when heart rate was slowest, patients with long QT3 mutations, sodium channel mutations, are at greatest risk of arrhythmia. And that turns out for most previously studied sodium channel mutations, due to the fact that this late sodium channel activity is more pronounced at slower heart rates than it is at faster heart rates. And so we asked whether or not in the iPS cells we could see a similar kind of effect for the sodium channel dysfunction the child was carrying, and whether or not drug application at a slightly higher heart rate might be more effective, and whether or not that was better or worse than using a second drug to reduce this dysfunctional late current. And what we were very happy to find was that the dysfunctional late current in the iPS cell sodium channels from the programs was very sensitive to slight changes in heart rate. It was greatly reduced the faster the heartbeat. And when you applied drugs at a faster heart rate, you got a much more effective inhibition of the late current with very little effect on peak current. And so the iPS cell readout was telling us that we could lower drug concentrations if we could increase heart rate, and we should see better control of arrhythmias in the proband. We needed to compare that with the possibility of using a second drug. Again, the consensus was to try flecainide. And what we had found when we looked at the addition of flecainide, and where we did get marginal changes in inhibition of late current, we found that mixilotine plus flecainide was really problematic in terms of inhibiting this potassium channel, which would contribute to prolonged QT. And so our recommendation for treatment was reduce the concentration, use mixilotine. It would be good at blocking late sodium current, which is the long QT signature form of sodium channel dysfunction causing the disease. Increase heart rate with the implantable ICD. And our prediction was that that would be a better way of controlling arrhythmias in the patient. And when that approach was taken, we looked at the if you will, the pre-change in clinical regimen data for the child, where these are the number of events that were recorded by the device that was implanted. And we found that we were able to control much more dramatically, much more effectively, exactly as we predicted from the iPS cells, by reducing the dose of the drug, presumably having that have a more specific effect on the sodium channel, reducing the off-target effects of the potassium channel, and even a slight change in the pacing rate through the ICD enabled a much better control of rhythm in the patient. And so, not perfect because these events were not completely controlled. This approach really gave us confidence that the use of iPS cells for this type of analysis, looking at normal and dysfunctional sodium channels in a disease where you have the clinical readout of the dysfunctional electrical activity, the ECG, you have the genetic data telling you which genes are affected, and you have the biophysical measures to tell you what the functional consequences of the gene mutations are. Using this approach to do in vitro pharmacology to optimize treatment for patients is really possible with iPS cells. And my hope is that this kind of study will stimulate other studies, better approaches to making iPS cells and turnaround quicker, looking at ways to screen drugs faster, and getting a quicker turnaround to patients. But we were very, very much encouraged by this approach. And so the conclusions are that iPS cell-derived myocytes have great potential at addressing mechanisms in drug therapies, particularly in cases with complex genetic backgrounds. There are often cases in long QT syndrome, for example, where not only are there polymorphisms in genes that accompany mutations in primary long QT genes, but there are mutations in more than one long QT gene. So that approach needs to be studied only really in iPS cells. An agreement between in vitro cellular pharmacology and improved clinical regimen provides strong support for unraveling drug mechanisms in patients with complex genetics. And the last slide is just a slide to tell you that I have a great group, very happy to have these people working with us. Rob Kass was the cardiologist who worked very closely with us on the case. John Banks and Minerva, Kevin, Wendy Chung, John Liu, John Liu's twice, Jenny, and Cecile Terrenoir and George Wang did the functional and pharmacological experiments in our lab. And Gordon Keller has been wonderful to collaborate with for the generation of the myocytes derived from the iPS cells. So thank you for your attention. I'm happy to take some questions.
Yeah.
Yes.
I was just curious, sorry, very technical point. Polarization of the, I guess, of the parental, again, this is on the sodium channel and on the single point mutation sodium channel. Did you notice a change in the tail current, the sort of the true deactivation kinetics of the channel? And could that explain the rate or the frequency dependence of the block? That's
a very good question. We didn't, in the absence of drug, we don't see any change in deactivation kinetics. But the stimulation rate changes the transition into this mode of gating where the channel reopens in a bursting way. And that's what allows the drugs to be preferentially inhibited there. It's a good question.
Are there any other questions? Running a few minutes late. Mark? You've been tutored long enough in this. You have to wait for the microphone.
So I'm curious about the context in which the potassium channel must reside in order for the drug to have the undesired effect on the potassium current. So you were unable to see that function when you moved to a heterologous, to a fibroblast or a non-myocyte system. So what is it about the cardiomyocyte that you think is important besides the genetics of having the sodium channel there and everything? Is there something specific about that environment that's critical for the drug to work?
So that's a really good question also. So because it was a quick talk and I went through things very quickly, this polymorphism in this potassium channel has been well studied. It's a very common polymorphism. And the literature is very mixed in terms, based on heterologous expression of whether there are functional consequences, whether there are pharmacological consequences or not. You can divide the papers 50-50. And so we felt it was really important to study this K channel in the environment of a cardiac myocyte because there are definitely other things that are important. Not shown. There are calcium channels, there are calcium handling proteins. There's a lot of other stuff that goes on in a myocyte that you don't get when you express things in a heterologous system. These are far from perfect. As anyone in the field knows, we're looking at early stage cardiac cells. They're embryonic. So there are key regulatory pathways that are at a very early stage of development, which are not going to be there probably. And there may be regulatory proteins that may or may not be there. But yet, if you look at the basic channels that we've been able to see, the sodium channel, the hERG channel that I showed you. I didn't show there's this other potassium channel that's responsible for long QT1. That's very prominent and very clear and very reproducible in terms of biophysical properties. So looking at pharmacology of those channels in this system, I believe is very good. Using the cells for other types of physiological, pathophysiological experiments needs more work. We need to have cells that are at a more mature developmental stage, for example, for many experiments that need to be done. But we definitely needed to have this K channel expressed within a cardiac cell to know whether or not we could believe there was something there or not there, because we couldn't rely on the heterologous system.